There we go. And welcome. So from genetics to psychiatry, uh, uh, and of course the gut microbiome, you study the fetal alcohol syndrome and how it's affected by the gut microbiome. And you do everything from lecturing to doing conferences and doing interviews like this. It's a bit of a super position, really, if you ask me. And so I was wondering, maybe could you tell me more about how you got to do this research? What led you to want to do this research that you're currently doing?
Yeah, so that's always a good question. I'm sure, like many people out there, I had a plan in high school. I was going to study a degree. I'm not sure what I was going to study. I was going to finish my degree at 21. I was going to get engaged at 23, married at 25, first kid at 27, and then at 29. I'm now 30, and we're still studying.
Oh yeah.
I haven't hit any of those milestones, and I think that's an important one. You know, you plan out this whole life journey, and then life takes you on its own roller coaster. So I think my journey really started. Obviously, I was at Stellenbosch, and then one of the buildings unfortunately had burned down. So we had our first genetics class in second year in the Kruiskerk, which felt a bit dodgy, because we were covering obviously some hectic evolution and that sort of thing in a church, but I absolutely fell in love with genetics.
Yeah.
And then I finished up my degree. I did an honors in genetics and then a master's as well. But my focus there, although I always wanted to do human genetics, the human genetics at the time was sort of on the back burner, and it was really important for me to do projects that develop as many skills as possible. You know, genetics, that's the joy of genetics. You know, everything, or nearly everything's got DNA. So as long as you have all the skills, you can apply it to everything. So then I did my honors and my master's, which was amazing. But then I needed a bit of a break. I wasn't quite ready to continue. And then I was really fortunate to get a job at the health science faculty at Tygerberg. There I started, or helped start up a lab in TB field. I had always thought, you know, it would be cool to like work for the CDC, you know, there's a new pandemic. Turns out it's not really for me. I learned a hell of a lot, but I also learned that infectious diseases is not something that floats my boat so well. But while I was there, I was exposed to the microbiome. And the one thing, I love genetics, but the problem with genetics is we can't do much about it. You know, once we're born, we're stuck with it.
That's what you have, yeah.
Exactly. So the fact that you could have bacteria specifically in your gut that you could maybe change a bit or modify, and that could actually affect your health, just blew my mind. And I started that sort of research in the TB field. So we were looking at the lung microbiome specifically, so how TB and other bacteria in the lung might affect TB outcome. But as I said, the infectious diseases wasn't for me. And then I sort of started looking to the mental health aspect, and specifically the neurodevelopment. So how those bacteria and their metabolites might affect brain development, and that's where it stuck.
Wow, that's quite a story, really. Wow. And so if you look at all the gut microbiome research that you're doing now, would you say, because initially you said that the genetics, you can't do anything about it. So are you saying that there's a sort of real world application when it comes to the gut microbiome that you and lung microbiome even, that you can change it?
So with the gut microbiome, for sure, you know, there's certain big things that affect your gut microbiome. The very first one is how you're born. That's unfortunately not something you can do much about, but obviously, hopefully your parents can do something about. And then the next big thing is what you are fed as an infant. So are you breastfed or not? Again, not really up to you. Sort of by the age of 2, 3, we have what's called the core microbiome, and that's able to buffer most changes. But we totally are able to modify the gut microbiome in sort of relatively easy ways, so just through lifestyle and diet, but then through more drastic ways. So one of the slightly disgusting sides of the field is a fecal microbiota transplant. So that's obviously only in extreme cases and shouldn't be done at home. And that's essentially, and it's been done in a few disease cohorts of particular interest to me in autism. Yeah. So where they essentially replace the gut microbiome, and that shows lasting effects on health. And in autism specifically, obviously it doesn't completely cure the child, but greatly decreases the symptoms, which is fantastic. So it's definitely a modifiable feature to human health, which I think is very exciting.
Damn, that's very impressive, actually. I wouldn't have thought that even something like autism, which is, from what I can gather, it's a complex disorder after all. That's very, very impressive. Now, yeah. I would assume, well, for me, something like that, like the fecal transplant, that would be a fairly challenging part of this type of research. Now, that leads me to ask, what would you say are the most challenging parts of the research? And maybe, in contrast to that, what's the most sort of favorite parts of your research?
Yeah. So obviously there are quite a few challenges, as I said, genetics, it doesn't change. So when you take a saliva or blood sample to extract DNA, that DNA doesn't change. Whether you're 1 year old or 50 years old, you're good to go. If I somehow lose your sample, I can go and, hey, can you give me some more? And it'll be the same. So that's one of the challenges in the microbiome. It's constantly changing.
Yeah.
So if you are doing, for instance, a longitudinal study, you miss a time point, you can't go back.
Yeah.
Also, just in the South African setting, one of the issues we have is actually dealing with that sample. So obviously, to study the gut microbiome, we need a stool sample. And the sort of gold standard is to freeze that stool sample immediately. And in the South African setting, that's just not an option. So we do have stool collection tubes, which are fantastic, and they mean that we can stabilize that sample for up to 3 months. But it means that we're compromising on some of the downstream applications.
Yeah.
So just getting around some of the logistics. There's also what one article deemed the yuck factor is always a bit of a challenge.
Yeah.
It takes a little bit more convincing to give a stool sample than a saliva sample. But in our context, we've been really lucky. The women that we work with are very happy to further this research. And then also because it's such a new field, especially on the bioinformatics side, things change at such a rapid pace. So, you'll spend 2 weeks doing a whole bunch of analyses, and in 2 months' time, like, oh wait, that stat's not appropriate. Let's use this one.
Yeah.
So because it's so fast paced, things that we did when we set up the study back in 2014, are not really what should be done now, but we have to make do.
Yeah.
And then my favorite thing to do, I love the lab work always, although it does have a certain yuck factor to it. And then I really like being able to share what I've learned, particularly like supervising students. So I have a really great student at the moment, and sharing that knowledge and helping them in their research, that's definitely a big one for me. And the conferences, traveling is great. COVID threw that under the bus a bit. The attending conferences and hearing other people's ideas is also a big one.
All right, so it's the teaching aspects and helping and sharing information. Does it become difficult sometimes to sort of balance doing your own research, but also then lecturing and teaching and going to conferences and traveling? Is it difficult to balance all of that?
So my lecturing load is very, very small, thankfully. I don't do too much lecturing, mainly to the honors students, a bit of basic genetics in the beginning and then some gut microbiome stuff towards the end. But yeah, for sure, it's always tricky to balance anything, doesn't matter what you do.
Yeah.
What I find works is don't procrastinate. get it done when you can do, and then prioritize. If you've got a deadline coming up, if that means everything else needs to be pushed to the wayside, then that's what needs to happen. But obviously, certain things come first. Like lab work. Lab work always comes first because that needs to get done first.
Yeah.
Writing is not my favorite thing, so I always try and push that to the bottom of the list.
Yeah.
But yeah, juggling is a constant thing. I don't just think in science. I think in life, it would be great to work linearly, but unfortunately, that's not the reality.
Yeah.
But we make it work.
Of course, yeah. I mean, who complains about traveling? We all, I wouldn't mind traveling a bit more, especially now after the COVID situation, like you said.
Yeah, I'm busy preparing for my first proper in-person conference since 2019, so I'm very excited.
Wow, yeah, it's been a while. It's been a minute. Which conference is it?
It's the Bio 2022, the FASD SAGS.
Oh, okay.
In Stellenbosch, yeah. So
Right,
I presented in 2018. I've presented some very preliminary results.
Mm-hmm.
So I'm excited to go back with proper results. It's exciting.
Okay. And is this part of because you form part of the FASD research group, is it as part of that now? Or
Yeah.
is it independent? All right.
Yeah. So it's on the, so I'll just give an overview of some of the research that we've done specifically in the gut microbiome. We also do a bit of the vaginal microbiome in Foetal Alcohol Spectrum Disorders, but that my student mainly handles very excellently.
Yeah.
But yeah, so we're just going to give an overview of some of the interesting findings that we found and how we're taking it forward.
And so what are some of the interesting findings that we found?
Yeah, so I mean, we've got 3 main parts to the study. So the first one was we want to look at the mothers. So we know that alcohol affects the gut microbiome, but we don't know how that is passed on to the infant. So our first thing was to look at the mothers. Do mothers who go on to have infants with FASD versus those whose infants don't have FASD, are there any gut microbiome differences? That data was less interesting than I was hoping for. I think the problem is we worked very closely with the FASD research team, who are fantastic, but they took the sample at any time point during pregnancy rather than a fixed time point. So unfortunately, the data was quite messy. During the 3rd trimester of pregnancy, there are big shifts in the gut microbiome. So I think that sort of clouded some of the results. But we found some very, very interesting results in the infants. So in this case, we looked at infants who were exposed to alcohol, those that went on to develop FASD versus those that didn't. And here, much to my relief, we found results relatively similar to autism, which is obviously a similar neurodevelopmental disease. So that made me feel a little bit more confident. The one quite strange thing is, so when babies are born, one of the first bacteria that they are exposed to is called the Phytobacterium. It's sort of a good indicator of health. It helps break down breast milk. So it's overall like a really good bacteria. And it has been shown in other disorders, including autism, to be decreased in infants with autism. And strangely, we found it to be increased in infants with FASD. So that's one of the things that we're looking into, because everyone I speak to is like, that's strange.
Yeah.
And we're not quite sure what the mechanism is of that. Yeah, so that's one of the things we're looking into. And there are other genera which have been associated, which play in nicely sort of to how babies' gut microbiome changes. So one of the things that we're also doing, just to sort of follow on that, is for the big cohort study, we looked at one time point at 9 months of age, which is around the time when most infants have been introduced to at least some solid food. So most of them are still being breastfed, but have a little bit of solid food. So it's when that microbiome changes happen. But we're also now looking at longitudinally at 6 weeks and 9 months. So obviously at 6 weeks, an infant is either completely formula fed or breastfed. Most of our infants are breastfed. And then obviously by 9 months it's changed. So is there persistent microbial difference between infants that develop FASD and those that don't? So we're busy analyzing that data now to see what we find.
That is very interesting. So you say that infants develop FASD. So would you say, is there a possibility that we could maybe treat them by targeting the gut microbiome before their development? Because does the phenotype only present later on? I always thought it was sort of immediately after birth.
So that's what we're looking into. So there are some infants that, if it seems like from birth, they're showing some of the morphological symptoms and all of that. But there are infants that are sort of borderline FASD, that sort of, as they get older, you start picking it up. And the sad truth is, the FASD team that we work closely with, they've been working in Robertson and Wellington area for over 25 years. And unfortunately, the prevalence of FASD just seems to be going up. It's not, even through all the healthcare, all the interventions, these women are still drinking. So it would be ideal if we were to find some sort of microbial change that we could either intervene with, or, my goal would be to, pop a probiotic, and then I don't think it will ever completely quote unquote cure FASD, but if it could just lessen some of those symptoms, then you sort of might be able to pull these children out of that circle of poverty. It's quite an issue. At the moment, we're looking at up to 31% of infants with some form of fetal alcohol spectrum disorder, which is shocking.
That's shocking, yeah. Is that in Robertson, or is that global statistics?
So that's specifically in the Robertson Wellington sort of wine area.
All right.
It sort of runs between 16 and 31%. Globally, it's less than 1%. So we're really overachieving.
Less than 1%, but with 9 billion people, that's still a few million people. Still a few million people. Wow.
Yeah, and the sad thing is that these infants, it's nothing they did. And then they generally don't achieve at school. They often have secondary comorbidities, and it's just this perpetuate this circle of poverty. And it's definitely something that if we could help at some way, some sort of intervention, particularly a microbial intervention, would be phenomenal.
Yeah, we need it. We definitely need it. Do you think, because you're obviously looking at it from the microbial perspective and microbial intervention, but do you think there are other intervention methods that we can also approach?
So there are quite a few interventions that are already being investigated and are in play. The biggest problem is identifying these children. Fetal alcohol spectrum disorder is, as the name says, a spectrum. So if the infant has full-blown FAS, you can see it in the face. It's got this morphology characteristics, not unlike Down syndrome. But then there are also infants that just have alcohol-related neurodevelopment disorders. So they're not showing any physical signs, but developmentally they're behind. So I think in the public school system, it's difficult to find these children. Intervention is tricky, but the FASD team has looked at a number of interventions, mother-child interventions, and then interventions with the children, especially later on in life, additional help as they sort of start entering the school system. But obviously all those are less biological.
Is there any, in terms of future research, maybe any other intervention method that you yourself would be interested in, apart from gut microbiome? Or are you set on the gut microbiome for now?
I'm putting all my eggs in the basket with the microbiome at the moment. So as I mentioned, we do also look at the vaginal microbiome. So most of our infants are born vaginally rather than C-section. There are 1 or 2 emergency C-sections. So obviously that is also, as I mentioned in the beginning, that's a big way that the microbiome is seeded. So if the mother has big shifts in the microbiome of a vaginal canal, then maybe that also affects the baby. But at the moment, I'm putting all my eggs in the basket with the microbiome.
And it's a good basket to put your eggs in.
I hope so. But I mean, obviously, maternal child interventions will always be great. And then, I mean, even if we could just do it, I know there've been some people looking at just dietary intervention. Obviously, that's sort of one step back from the microbiome. So it could actually be essentially what they're doing is altering the microbiome without actually knowing it. And there have been some interesting studies done in that. But it's because it's not a huge burden elsewhere, it doesn't receive the attention that it probably ought to. But there are lots of people looking at neurodevelopmental disorders in general, which is great.
Of course. Would you, if an undergraduate student comes to you now and says they're also interested in, studying the gut microbiome, what pathway do they follow? How do they get to where you are?
That's a good question that I can't answer. Everyone's journey is different. I can't tell you how to get where I am because how I got here is my way and everyone else has their own. But I think if you're passionate enough and you find the right people to be in your support system, they can help you along the way. It's not a sole journey. I couldn't have got to where I am today without my supervisors, my family, all the support around me. And then, just the opportunities. So jump at every opportunity that you get, even if you're unsure, if you're not sure, apply for that thing. The worst they can do is say no. That's what I do all the time when I want money. Might as well try. So I mean, I can't tell you how to get there. But if you've got the passion and you surround yourself with the right people, I think you'll find your way.
That's a great answer. That's a really, really great answer. Thanks. And now, if you weren't studying the gut microbiome, what else do you think you would have found yourself studying? So
that's, the one thing that I love and hate about myself. A lot of people, when they're finishing up school, what do I study? This, that. I have always been incredibly one-track minded. Always want to do something in the biological sciences. So I don't think I would have deviated too far. I dabbled in some physiology and anatomy, and none of that really worked for me. So I think I would honestly stay in the genetics field, maybe some more typical genetics. I love, as I said, the genetics is very important, but it's not really changeable. So I think if I were to look at another field, I would again go for something that might be slightly modifiable. So, some of your epigenetic regulation. I find some of the DNA methylation work that's going on very interesting. So that's probably where I'd be at. One track minded helps at sometimes.
I mean, you know what you want, so that's good.
Exactly.
Do you think some of your future research might include some epigenetic or different methylation patterns or anything like that?
I hope so. So some of the interesting work that's being done is sort of involving the microbiome with other fields of genomics. So you can do a microbiome genome-wide association. So look at what genetic SNPs or genetic changes are associated with different microbial genera, or the interplay between the microbiome and DNA methylation. So I think that would be very interesting to look into, sort of as another level to just the basic microbiome work.
Wow.
And then as I mentioned, I mean, I was also interested to do functional work, but that's where the issue comes in again, going back to the sample collection, the way that we collected our samples in the sample collection tube. It allows us to store the sample at room temperature for 3 months, but it lyses all the cells, so we can't do functional work. So if we could go back and re-sample some of them in sort of a different timeline, obviously it would be cool to do some functional work. And I think it's really important going forward. You know, for a long time there's a big focus on what bacteria are in the gut, but more and more we're looking at what are they doing in the gut.
All right.
You know? So I think that would definitely be interesting to look at.
Future prospects, yes.
Yeah,
wow.
I've got a list.
You've got a list. Well, Natasha, I'm looking forward to seeing the results of your PhD study. I'm looking forward to hearing about all the different applications, and then hopefully I'm also hoping that all your eggs in the basket of being able to treat the spectrum disorder really works out, because it's really some amazing research that you do. And honestly, it's been a pleasure talking to you today, and thank you so much for taking time to talk to it.
Thank you so much for having me.
Yeah. All right.
Keep well.
Okay. Thank you, Natasha. Goodbye.
Bye.